So Devin, I know you'll be projecting the screen today.
Yes, should I go ahead and do that now?
No, I'm gonna actually just throw up a quick title slide just so that everybody knows
good.
where we are while we, I just wanna make sure that the settings are correct. So Devin, I know you'll be, okay. I'm sorry, this is why I just wanted to make sure it was all set up before. Luckily, you can pause the Zoom recording. So I'm gonna just start off sharing the screen. I know that we are officially at the top of the hour, and Devin, I'm gonna hand it over to you in just a second, but we are very excited today to have this call. And just to provide a bit of background, the Research Literacy Networking Zone is something that has been a tradition now since 2016 at AIDS conferences and the Research for Prevention conferences that happen in alternate years, as well as other meetings that pop up around the globe. And the point of the zone is when people come together for these conferences to share experiences and to share knowledge, this zone provides a space to really break that knowledge down for advocates and community. And unfortunately, with COVID-19, we're not able to be together in person, but perhaps a silver lining is that it really helps us expand this out globally and reach more people through this promotion of online platforms. So this is a pop-up series that we'll be doing throughout the week. And this one is on broadly neutralizing antibodies. And we're really excited and very lucky to be joined by two incredibly smart, incredibly talented individuals from IAVI, Olinka Fagabi, but I'm sorry, Olianka Fagabi.
It's hard.
Who is the program director of IAVI Neutralizing Antibodies Center. And she's the primary point of contact for the NAC. And Olianka works with internal and external stakeholders to oversee and ensure the coordination, administration, and implementation of IAVI's Neutralizing Antibodies Center to define and execute the high impact scientific activities at the NAC. And Olianka serves as a resource and interface for the executive design and delivery of the scientific, programmatic, and organizational vision. She earned her bachelor's degree in biology with a research focus in glycobiology from Stanford University. We're also joined today by Devin Sock, who is the Senior Director of Antibody Discovery and Development at IAVI. Devin is IAVI based at Scripps Research. His group is currently focused on antibody discovery and development for HIV, as well as other disease areas, including snakebite and antimicrobial resistance. Devin is generally interested in the mechanisms of antibody-mediated protection against infectious diseases and antibody production development to prevent or treat neglected diseases that affect those in low and middle income countries. So I'm going to let him share the screen to take over the slides, but we do encourage people to ask questions in the chat, and I'll also be taking some questions on the YouTube channel as well. So go ahead and ask questions in either location. Thank you.
And I'm going to just kick it off here, just cut in for Devin. Hi, everybody. Good morning, good afternoon, good evening. Very excited to be joining today in this session. Again, my name is Olinka Fagbai, and I serve as the program director at IAVI's Neutralizing Antibodies Center, and that's on Scripps Research Campus in La Jolla, California. And I'm joined by Devin here, who is our Senior Director of Antibody Discovery and Development. We really appreciate IAS AIDS 2020 to create this platform, and thank you AVAC for bringing this together and letting us join in on this session. So today we're going to be discussing a bit about scenario planning for the AMP results. And AMP is an antibody-mediated prevention study, and it's really testing whether a specific class of antibodies, VRC01, can protect against HIV through IV infusion. But what I'm going to do today, before I pass it on to Devin, who's going to provide a bit about the AMP results and the science at a really good level of detail, is give some general background around antibodies. Next slide. So as general background, what are antibodies? So antibodies are proteins that are made by our immune systems in response to infection. So in the case of HIV antibodies, they bind what's called the HIV envelope trimer, and that helps prevent HIV from infecting target cells. So on this slide, HIV is really shown at the bottom, and all coated along the surface, sparsely decorated, are these envelope trimers, which then bind to the target T cell, which is shown right on top. And that CD4 and CCR5, those are the things that really enable the fusion and entry of the virus. So antibodies really are the block, and they're essential for blocking the process of binding those, the envelope trimer spike proteins, and essentially block infection. So when we call antibodies neutralizing, and you'll hear the term neutralizing, they're really saying that they're blocking an infectious agent from interacting with the host cell. So another term, another term you guys will probably hear often are broadly neutralizing antibodies. And those are characterized by the ability of an antibody to neutralize multiple global isolates of HIV or variations of HIV. So broadly neutralizing antibodies, they typically, in natural infection, they develop very late in infection after individuals are exposed to repeated strains of HIV. So other terms, we typically, from an HIV vaccine perspective, are trying to elicit or get our body to create broadly neutralizing antibodies. And additional terms that you'll hear beyond broad are also terms such as potent. So I associate potency with how tightly an antibody can bind to its target. So the more potent something is, the less that you actually need to block infection. Whereas again, when antibodies are broad, they don't block just one strain of HIV, but they can neutralize a diversity of different antigen targets and HIV variations. So in terms of what antibodies bind, when we're talking specifically about HIV, there's different classes on that HIV trimer spike that's shown at the bottom. They target different groups of sites, and those different sites are the V2 apex. I suppose if my head was an HIV trimer, it'd be the crown on top. We have the V3 glycan. The CD4 binding site might be around here. We have the fusion that's at the bottom, and then the emperor that's also at the bottom. And there's been a whole host of different antibodies that have been discovered against all these different sites. And what's really great about what scientists have done to isolate these antibodies is that they're used as tools to help us in making a vaccine, because that's ultimately what we want our body to create. But as antibodies have become more potent and more broad, they also can be used as therapeutic agents in and of themselves for both prevention and treatment. And antibodies are not only things that we're thinking about in the case of HIV, they're existing antibody drugs on the market, and they typically are safe, they're long acting, they're potent, so they're really great potential therapeutics against disease. But the barrier typically for antibodies to be accessible to everyone are that there's barriers in terms of affordability and accessibility, which is something that we're focused specifically on at Alvia, and Devin's going to touch on a bit more in his part of the presentation. So a little bit of that background about what antibodies are. I'm going to have Devin get into a bit about the AMP results, the AMP study, and why we're excited about it.
Great, thanks Olayinka, and thanks Jessica for the introduction. So I'm going to be describing, building off of what Olayinka just described in terms of what are antibodies and what is neutralization, how do they work, and how does this all connect to AMP, and why are we so excited about AMP. So this is a slide that summarizes kind of the context for why AMP is so important and really historic. So in terms of what we know, we've done a lot of experiments in non-human primates. So we've taken antibodies that are neutralizing. So as Olayinka described, antibodies that prevent HIV from infecting target cells. We've given these antibodies to rhesus macaques, and then we've tried to challenge these monkeys with a version of HIV that can infect them. And we give them through different routes, either intrarectal, vaginal, intravenous, a variety of routes. And what we've seen is that if the antibody is capable of neutralizing and is present at a certain concentration, that those animals are completely protected. There's no detection of viremia in those monkeys. And we've done these experiments for the past decade, and we've used a number of different antibodies, we use a number of different viruses, and we've given the antibody in a number of different doses. And so why is that important? So really it's highlighted here in this graph. And what you can see is that each of these dots represent a different study where a different antibody, these are color-coded by different epitopes, which is what Olenka described in terms of the V2 apex, CD4 binding site, et cetera. Each dot represents a different antibody. The colors represent different epitopes, and there are different challenge viruses that are given, and these antibodies are given at different doses. And so what we know is that on the y-axis measures the amount of antibody that you can measure within the serum of the monkey. So once you give the antibody, how much is present in the serum? That's depicted on the y-axis. On the x-axis kind of demonstrates what we measure in vitro. So what we measure in the lab in terms of the amount of antibody that you need to prevent virus from infecting cells in a petri dish. And what we see is that there is a correlation. So it looks like these series of dots form a line, which indicate that there's a pretty strong correlation of the potency that we measure in vitro in a petri dish compared to the amount of antibody that you need to protect against HIV in a non-human primate animal rhesus macaque model. What we also know based on these data and all these different experiments testing these different antibodies at different concentrations is that the more potent the antibody is, the less that you have to give in order to get protection. So you can see the highest dose in this graph is 40 mg per kg, which is a huge amount compared to 0.08 mg per kg. And that's a huge difference in amount of antibody that you can give and still get protection from HIV. And so why is this important and how does this link to AMP? So AMP is the antibody-mediated protection study. All the work that we've done so far have been in monkeys. And of course, our objective as scientists, as policymakers, as advocates, as people working on HIV, is not to protect monkeys from HIV, it's to protect humans from HIV. And so what we're trying to do is now that we have all these data that we've generated in monkeys, where we know that antibodies are capable of protecting against infection, what is the corresponding concentration that's required to protect humans from HIV infection in the field? And so one of these antibodies, and that's this green one here, VRCL1, is the same antibody that we've tested in monkeys that were also given to people to protect against real-life infection, real-life exposure from HIV. And what we're going to understand is how much antibody is required to protect people from HIV infection, how well these animal models actually predict what we expect to see in people. And really this information is going to be incredibly important for vaccine design. So how much antibody do you need to elicit in order to get protection, as well as important for the design of new antibodies that are more potent to figure out how much you need to give, how low can you go in order to still get protection. And we care about that because we need to be able to increase the affordability of these antibodies. So if you can give less, if you can get away with giving less, the cost of that product will go down dramatically. And so that could increase the number of people who can benefit from that intervention. There are a number of different outcomes from the AMP study. So this is the first experiment where we're going to be where we've delivered a neutralizing antibody to people. This is delivered both in South Africa as well as in the Americas among women as well as men who have sex with men, given at different concentrations, and they're followed over time. And so there's a number of scenarios of what we expect to see. And so the worst case scenario is that we see absolutely no protection. This would go against everything that we've generated in the lab and would be a huge impact to the HIV field. The possibility, in my opinion, is going to be very unlikely that that's the case, but it's still not a 0% possibility. So that's on one extreme end. The AMP study says that antibodies are not protective. On the other end, we see that the way that the study is designed is that these antibodies, the VRCL1 that's given, isn't 100% protective. We don't know, just like any other HIV prevention product, there is a threshold of predicted efficacy for that product, depending on how it's used and how it's administered and the concentrations. And so the best case scenario is that we see complete protection across the board. Even in cases where we don't expect to see a huge level of protection, we see protection everywhere. So those are the two extremes: no protection, complete protection. What's fuzzy is somewhere in the middle. So at some point, we're going to see whether or not there's modest levels of protection or poor protection. And the way that this study is designed is that for those who do become infected after given this antibody, we're also sequencing the virus. So we're going to see what the virus looks like. We're going to see whether or not the virus is sensitive to the antibody. And we can use that information to really figure out what is this scenario 2 versus scenario 3 situation. In effect, what we're trying to determine is it an issue of breadth? Do you need multiple antibodies for every virus that you're exposed to? Do you need multiple antibodies that can neutralize that virus so you have multiple layers of protection? Or does it have to do with potency? And those things, of course, are linked, where you might not need necessarily duplicate coverage, but you definitely need potent antibodies to protect against infection. And those two different scenarios are a little bit more fuzzy, and we can go through them in the question when we open it up for questions. I just want to do a quick plug in terms of the work that we're doing as IAVI as part of our collaboration with the NIH at the VRC and what we're working towards. So as I mentioned, what we care about is making sure that it's not enough to have an antibody and show that antibodies are protective. What we really need to do is make sure that the antibodies are affordable, available, and accessible to those who really can benefit from them. One of the biggest issues for antibodies for use for prevention is their costs. So a lot of antibody therapeutics that are being used in cancer, these are not costs of goods, of course, these are overall costs. They're incredibly expensive. So in the US, they can go up to $200,000 per year for treatment compared to antiretrovirals, which are much more affordable. So one of the big questions is, how do we bridge this affordability gap? How do we make sure that if the AMP studies are really positive, we know that antibodies protect, how do we make them affordable so that more people can have access to them? And so what we're doing at IAVI as part of our collaboration with the NIH is focusing on these three things. So how do we improve the potency of the antibodies so you don't have to give as much and get protection? How do we increase the lifetime of the antibody? So if you can give it once and have it last for at least 6 months, 3 months, 6 months, even 9 months, hopefully a year, you can reduce the number of times you have to get the antibody, and that will overall reduce the cost of getting that intervention. And then finally, can we partner with people in LMICs that are capable of low-cost manufacturing to further reduce the cost of goods, the COGS? We think that a combination of these three different activities will effectively reduce the cost of antibodies and make an HIV antibody product available for low and middle income countries. So this is just a summary of where our antibodies are. So we've been working on optimizing these antibodies for enhanced potency, as well as our collaborators at the NIH. And these are kind of where the antibodies sit in relation to the AMP study. So you can see the AMP study in green versus our antibodies in red and blue. The way to read this graph is the farther left these antibodies are, the more potent they are, the farther up they are, the more broad they are. And so you can see as our next generation antibodies that we're working on, that we have better antibodies, they're more potent, they're more broad. If the AMP study is really positive, we think that what we have in the pipeline is going to go above and beyond the antibody that's been tested in the AMP study and potentially enable broad access to what could be a really effective product. I'll pause there. I think this is the end of our slides, and just open it up for general questions from everyone who's online. Jessica, I think you're on mute.
I am on mute. Thank you. So Devin and Olianka, that was a wonderful overview of where we are. So a couple of questions have really started to come in. I'll start with one. So Devin, you actually presented a slide on sort of the scenarios of what happens if AMP is sort of, if we see no results, if it's flat, or if it's very successful, and then this sort of two in between. And the two in between showing a combination approach, or what looked like it could be a combination approach of antibodies. One of the questions is why not start there? Some data suggests that a combination of antibodies actually might be the way to go. So why not start with scenario 2 or 3 and put that together and move that forward, regardless of sort of where the results of what the results of AMP end up being?
Yeah, it's a good question. So there's a combination of things. So for just speaking to our antibody development program, uh, we are focused on combinations. So the way that we've selected the antibodies that we are advancing as a affordable product is responsive to whatever the results are going to be from AMP, either it be a really good scenario or a less good scenario. We have combinations that we're advancing in anticipation that we probably will need combinations. What's important though is if scenario 4 happens, where VRC01 blows it out of the park and AMP is a resounding success, then there's a huge amount of benefit to having a single antibody. So it simplifies how you can deliver the antibody. It enables different routes of delivery of the antibody. So instead of having to sit with an IV drip for 2 hours, you can just get a simple shot. It reduces the costs dramatically. So instead of having to produce and combine 3 combinations of antibodies, you just have a single antibody. So there's a huge amount of pros if scenario 4 is the case, and we are all crossing our fingers that that is the case. But we're also prepared for what is potentially more realistic, that you need a combination of products, a combination of antibodies. But the scenario 2 and 3, as you're listed here, is an attempt to simplify what's going to be a very complicated analysis. We're going to measure what concentration of the antibody is in these individuals when they become exposed, as well as what the virus looks like. And there's a lot of nuance and details in how to figure out what that dynamic looks like across all the different individuals, across the different demographics, across the different routes of transmission, to really discern and conclude as best we can what scenario 2 and 3 look like and what we think we need for a future product to be effective.
I mean, that's a really helpful explanation. And I think that leads into the next question really well, which is, can age, region, and differences in genetic makeup interfere with how antibodies work?
It's a really good question. I will say that those particular attributes, and this is part of the promise of antibodies, those particular attributes are incredibly important for vaccines. I want to first iterate that vaccines work, but we do know that given different demographics, where you are, your age, your health history, your response to vaccines can vary, and that could impact the efficacy of the vaccine. The power of antibodies, as we've described them, and delivery of antibodies, both for prevention and for therapy, is that you can control it. So it's the same antibody that you give regardless of your demographic, regardless of your age, regardless of your situation, and it should have the same effect. It's a much more tunable control of the immune system versus a vaccine. But a vaccine, of course, is still very much important and still the priority.
And I just want to add something that's really important about what we're doing in terms of the antibody studies as it relates to the vaccines, is that what a vaccine is trying to do is actually elicit from your body this type of antibody response. So what we learn, it's actually complementary and synergistic of what we're doing, both from a vaccine perspective to elicit these broadly neutralizing antibodies or delivering them for prevention or therapy.
That's great. And I mean, for those who are attending AIDS 2020, Tumbi Ndugu in his plenary talk yesterday really stressed that idea of the need for a vaccine and sort of began to talk about some of these issues. So another question that is coming in are there vaccines or medicines that can induce these broadly neutralizing antibodies, or are they always going to be an infusion?
I think that is the $1 million question. That's the holy grail, is, and a huge number of people around the world, including Thumbi, are working on this in terms of how do you elicit these broadly neutralizing antibodies. So what we know is that these antibodies are special. They're capable of neutralizing a diversity of HIV. We know that a lot of them have been isolated from people naturally who've been infected with HIV for a long period of time. And we also know that they have weird features. They don't, because they've been isolated latent to infection, they have acquired special features that have enabled them to broadly neutralize HIV. And so that makes it more complicated for vaccine design, because you have to come up with really clever techniques, clever designs, clever immunogens, clever immunization sequences to elicit these really special antibodies. And so huge amount of effort from a lot of people around the world. Looks like there is some promising data, and hopefully that will be discussed relatively soon in terms of clinical trial results, but a huge effort from the field.
Great. So we actually have a lot of questions coming in sort of a variety of platforms. And one is, what are your thoughts on the new Cabala result? And does that impact how you're thinking about broadly neutralizing antibodies or life after AMP now that we do have HPTN 083, the HIV Prevention Trials Network, results in MSM and transgender women?
Yeah, so it's a very good question. So the CAB results are incredibly exciting. I think they're very promising. But I guess the role of antibodies, the CAB integravirs, the current administration is IM injections in the butt. And so we know that there's probably going to be, as we know throughout the history of HIV prevention research, differences in preferences, right? So we do think that Cabala is going to make hopefully a really big impact in terms of HIV incidence and prevalence, but there's likely going to be a need for additional products that have different ways of administering them, self-injection, for example, subcutaneous injection, that provide different intervals of protection, be it 3 months, 6 months, etc. And so it's part of the whole concept of having multiple prevention options is going to be really important to really drive down the epidemic.
Great. So, that really leads us into our next question, which is, we have the results from the MSM transgender women trial for Cabala. We're still waiting for women. And one of our questions is, do you think there might be a difference in the results between the two AMP trials happening now? So do you think there might be a difference in the way MSM and transgender women versus cisgender women actually have different antibody results?
I'm loving all these questions. That's a very, very good question. I think I do think that the study was designed to test that. So we can see based on women in South Africa versus MSM in the Americas whether or not there is a difference in protection. And there's multiple layers. There's different differences in the viruses that are circulating in those regions. There's an assumption in terms of the route of transmission, et cetera, et cetera. But there are some things that we can potentially learn from protection efficacy. What we know, at least based on recent macaque studies, is that we get protection regardless of the infection route. So we get protection if you challenge with virus intravaginally, intrarectally, or intravenously. So we don't anticipate that there's going to be a difference if the concentration of antibody is present at an amount that will protect against the virus. And that's the really key part. What is the amount of antibody that you need for protection?
Yeah. So we just have a few minutes left, and I know that there are going to be questions that we don't get to, but this question has actually come up in a couple of different forums, which is, what are your thoughts on how these broadly neutralizing antibodies compare to the SARS-CoV-2 antibodies that we're hearing a lot about in the news?
Yeah, it's also a very, very good question. We are also we are also involved in SARS-CoV-2 neutralizing antibody work as well. I think the important thing to really highlight is, and I think also the importance of continuing to fund HIV research at all levels, from advocates to scientists to policymakers, everything about HIV vaccine research, because that work has really catapulted, expedited all the work around SARS-CoV-2. And that includes the isolation of those neutralizing antibodies to a lot of the vaccine design work. And so I think, so just wanted to say that because I think it's really important in terms of the value of the collateral benefit as we continue to work on HIV work. But as to the antibodies themselves, the antibodies that we've isolated from SARS-CoV-2 are incredibly potent. A lot of the groups that have reported them are exceptionally potent, as potent as some of the most potent antibodies for HIV. What's interesting is that for SARS-CoV-2, it's an easier virus. It's a single virus compared to the diversity of HIV. And it seems like you don't need very much to elicit those really good antibodies. So it looks like the antibodies that we pulled out that are incredibly potent look very similar to, aren't very mutated at all. They don't look like those special broadly neutralizing antibodies. So huge difference, very similar levels of potency.
Great. I know we're getting to the top of the hour, and there is a question about sort of the AMP trial specifically, which we won't answer, but I think it raises a bigger question, which is, as you move forward after AMP, but just in general, how do you select the combination of antibodies that go together for a clinical trial?
It's a very good question. So for us, for our development program, there's two aspects. So, and Ole could describe them in the introduction. It's breadth and potency and safety. So breadth in terms of there's these different antibodies that target different epitopes, have different coverage. So CD4 binding site antibodies, for example, have very high coverage. They neutralize over 90% of viruses. Antibodies that target to the V2 apex and V3 glycan, for example, have limited, comparatively less breadth. They're more like 60% broad. They can recognize 60% of virus isolates, but they are way more potent. So they're typically much more potent than CD4 binding site antibodies. And so in terms of selecting the different combinations, the way that we've structured this and the way we've selected our antibodies for our combination is, if we select two antibodies that target the most potent epitopes, V3 glycan and V2 apex, what we know is that the combination of an antibody against V3 glycan and V2 apex is really, really broad. So we have the best of both worlds. We have super potent antibodies that together are very broad, but then we also want an added layer of protection. So we add a CD4 binding site that is also very broad. And so that's part of the thinking around it. And of course, it's more complicated because we need to match, the three antibodies. If it's a 3 antibody cocktail, we need to make sure that they all last as long similarly in the body when you give them in terms of how long they last, that they are given at the right doses because they have different levels of potency. So there's a little bit of a nuance there, but effectively we want to emphasize breadth and we want to emphasize potency.
Great. So I know we're at the top of the hour, but it came up in our last session, so I'll say it here. You've made the science that's so complicated seem so easy. So I don't know how you both did that, but you really brought it down, and I know that I really benefited from the conversation today. Actually, there's one question that maybe we can ask before you guys go. Can BNAbs work in a population for a long time? And is there a possibility for resistance? Because I actually think this is a good piece for people to know.
Yeah, that's a really excellent question. So my honest answer is we don't know. My thoughtful answer is it's going to be relatively unlikely. So the challenge with CAB, for example, is that it's given as both a therapeutic and as a prevention. And so when you combine them, and the antibodies are also being thought of as both for prevention and therapy. You select for escape viruses whenever you give anything as a therapeutic, because you have an enormous amount of virus diversity within an individual that if you give an intervention, you can always select for escape. In the case of protection, though, based on the data that we know, the diversity of, when you're exposed to HIV, it's not a big diversity of viruses that establish new infections in an individual. As best as we know, based on the data that's available, it's likely 1 or 2 specific types of viruses within that larger swarm of viruses that really set up a new infection in a new person. And so we really don't necessarily have to protect against all the diversity of viruses within a given individual in chronic infection. What we really need to protect against are those couples of viruses that establish infection. And we call those the transmitted founder viruses. And so, in my opinion, and of course this needs to be borne out by data as we administer these antibodies over time, is that the barrier to protection is going to be much lower than the barrier to therapy. The being able to protect against 1 or 2 viruses that seed infection, it's going to be much easier than protecting against a huge diversity of viruses in someone who's been infected with HIV for a long period of time. But as anyone who's been involved in HIV for however long knows, escape always happens. And it's something that we need to be conscious of. It's something that we need to be worried about and thoughtful of. If antibodies become a prevention product for the next 30 years, what is the virus diversity going to look like 30 years from now? And we are, as a group, already thinking about that because that is relevant, as we all know, with HIV.
Great. So I just want to thank you both so much for your time and your expertise and your knowledge to share with us. We will be having just, continuing this conversation, trying to gather the questions that came in through the Q&A and sharing those with everybody. But just thank you both so much for your time. And if you're interested in continuing this session, I will just do a shameless plug. On Friday, we're actually going to have Shelley Karuna talk about the antibody-mediated trial as well. So we can dive into this topic again further on Friday for those interested. But thank you so much, Devin. Thank you so much, Ulianka, for your time and your expertise today.
Thank you so much.
Thank you, Jessica. Thanks, everyone.
Have a great day.
Ciao.
Bye-bye.